Have you ever heard the story of the ship of Theseus? So the story goes that Theseus saves the children of Athens, and as a result, every year the Athenians would commemorate this by undertaking a pilgrimage. And of course, every time they returned from the pilgrimage, the ship would need some maintenance, maybe a plank replaced here or there. The thing is, after many, many years of pilgrimage and maintenance, the people realized that there was nothing of the original ship left. Every plank, the mast, the sails, all of it had been replaced, which led people to ask, well, is it the same ship? Now, I know what you're thinking. Either you've inadvertently stumbled upon the incorrect video, or I'm kind of having a bit of a philosophical crisis here about the persistence of identity. Well, yeah, maybe, but our bodies do the exact same thing as those maintenance crews on the ship of Theseus. We replace thousands upon thousands of our cells every single day. And given that we know that cells can only come from pre-existing cells, what this means is that throughout our lifetimes, many of our cell types, though interestingly not all of them, but many of them will regularly be undertaking cell division to produce new cells for the purposes of growth when we're younger, but when we're older, well, for maintenance. And you see, the thing is, any replacement cell needs to be able to carry out the same functions, have the same activity as the cell that it replaces. And what we know, what enables function of cells is DNA. All cells, all life, in fact, uses DNA as its information system, and it all uses it in exactly the same way. The DNA contains the codes, or the instructions, if you like, for building proteins, and it's those proteins that actually direct the activity, direct the functions of the cell. So if we think about this, what this means is that the key to the continuity of life is that every time a cell is going to divide, it needs to make a copy of that DNA so that when it eventually splits, both of those resulting daughter cells have all the information that they need to function. This copying of DNA is referred to as DNA replication, and DNA replication is the subject of today's video. Put very simply, without DNA replication, we can't have cell division, and without cell division, well, we can't grow or repair our bodies or maintain them, as we said. Importantly, DNA possesses two key elements in its structure that are absolutely integral to this process of DNA replication. They are complementary base pairing, and then secondly, the orientation of the strands. So let's just take a minute here to review what we're talking about. If we look in the nucleus of any of our cells, we can see that each of our DNA molecules, or chromosomes, consists of two polynucleotide chains wrapped around each other in a double helix. Well, those two chains, though, are held together by bonds between the bases of the nucleotides that swivel to the interior of the helix. But what is so important is the very specific nature of the relationship of that bonding. What we're saying here is that adenine always and only bonds to thymine and vice versa, and that cytosine always and only bonds with guanine, and again, vice versa. This is complementary base pairing. What it means is that if we look at our double helix, but we only happen to look at the sequence on one of the strands, we still know unequivocally what the sequence is on the other. Each strand is complementary to the other, and this is absolutely essential for the mechanics of replication. But hold that thought for a moment, because the other all-important structural property of DNA is the orientation of the strands. Now, if we look at any sequence of nucleotides, what we will see is that at one end there is a phosphate group, and at the other end there is a hydroxyl group. Very simply, this means that we can identify one end from the other. Now, the end with the phosphate group on we refer to as the 5' end. Very simply, because the phosphate group is attached to the 5' carbon of the sugar of the nucleotide. The end with the hydroxyl group we call the 3' end. Why? Well, because that hydroxyl group is bonded to the 3' carbon of the sugar. So what this means again then is that any sequence of nucleotides has a 5' end and a 3' end. In other words, there is a sense of directionality to it. Now, when we look at a DNA molecule, we are looking at two polynucleotide chains wrapped around each other. And interestingly, if we go out to the very end of the molecule, we'll see that the 5' end of one chain sits opposite the 3' end of the other. And then if we come back down to the other end, we see the same, just flipped. What we're saying is that the strands are running in opposite directions, and we refer to this as anti-parallel. Now, bearing those two things in mind, complementary base pairing and the anti-parallel orientation of the strands, let's actually take a look at a cell that's going to divide. The important phase of the cell cycle during which DNA replication occurs is called the S phase, or synthesis phase. And it's very important for us to appreciate that this whole process of DNA replication requires multiple large protein complexes to actually make this occur. What we're going to do is really just look at some of the main players and see how they all contribute to copying of this DNA. And the first group of proteins is all important because we need this to actually separate the strands of the DNA so that we can carry out replication. Now, this happens at something called an origin of replication. Very simply, an origin of replication is just a sequence in the DNA. A sequence in the DNA that this complex of proteins is able to recognize and attach themselves to and start the process of separating the strands. Now, just a quick kind of point of interest here. As eukaryotes, our chromosomes are all linear. And in fact, on every single one of those chromosomes, we have multiple origins of replication. So that is to say then that replication is being initiated actually at multiple points along the chromosome at the same time. But back to this first group of proteins, because chief amongst them is a protein called a helicase. And a helicase is a type of enzyme that is able to untwist or to separate two polynucleotide chains. It actually does it because it's able to just break through the hydrogen bonds. And so when the helicase separates these chains, what it creates is what we call a replication bubble. And at the ends of the bubble are a Y-shaped region that we refer to as the replication fork. And once those strands have been separated, the helicase is then going to go to work at the point of that replication fork, and it's simply going to move along the helix, continuing to separate the strands. Now, assisting with this are something called single-strand binding proteins. Now, as their name suggests, they bind to single-stranded DNA, so strands that have been separated. Why do we need these? Well, it's actually two pretty interesting purposes. The first is actually it prevents degradation. So as it turns out, single-stranded DNA, meaning when you've had a helix and you pull the strands apart, is more vulnerable to breakdown. So the single-strand binding proteins help to prevent that. The other cool thing that they do is they actually prevent the strands from coming back together. So what I mean is this: if we just left the helicase on its own, it would continue moving along the helix, pushing its way between these two strands and breaking the hydrogen bonds. But the thing is, if that's all that happened, those two strands would come back together behind it. It's called re-annealing. Well, those single-strand binding proteins stop that from happening. They stop the two strands coming back together and so help to keep them separate so that the actual process of synthesizing new DNA can occur. But there's one more protein amongst this complex that I want to bring our attention to, and that's called the topoisomerase. This is maybe the coolest of the bunch. So what does it do, and why do we need it? Well, as that helicase is moving along the DNA helix, breaking the hydrogen bonds, separating the strands, what this creates ahead of it is what we call overwinding. It's also referred to as supercoiling, but that's not a good thing. It's the topoisomerase that deals with this. As the overwinding starts to occur, as too much tension starts to build up within the double helix, the topoisomerase makes a small nick, a small cut in the DNA, and what this does is it relieves that overwinding. And it then joins up that little nick again. So between the actions of the helicase, the single-strand binding proteins, and the topoisomerase, our DNA is now ready to be replicated. And this is where now we start talking about our second all-important protein complex, those that will actually do the work of synthesis. First one I want to point out to you is something called a primase. A primase is an RNA polymerase. Now, that might cause you to stop for a second because we're talking about DNA synthesis here. Why are we talking about an RNA polymerase? An RNA polymerase builds chains of RNA. It puts RNA nucleotides together in sequences. The reason we're talking about primase, the reason we need it, is because the big enzyme that does most of the work of DNA synthesis is called DNA polymerase III. But it has a couple of significant limitations. Firstly, DNA polymerase III cannot initiate DNA synthesis. It actually needs a pre-existing chain of nucleic acid or chain of nucleotides as its starting point. The second important limitation here is that DNA polymerase III, even when it has that starting chain of nucleotides, can still only add onto the 3' end. So what we're saying is that you can separate the strands, you can expose the DNA, but the DNA polymerase is not able to simply bind to that single-strand DNA and start to synthesize. What it requires is a primer. It requires a short 5 to 10 nucleotide sequence of RNA that it can use as its starting point. So what the primase does, because it is able to start synthesis from scratch, is it is able to bind onto that exposed DNA, produce a 5 to 10 nucleotide primer, and then our DNA polymerase III is able to latch onto the end of that and start synthesizing. And of course, what it is doing is it is building a complementary chain. It uses the original, or as we call it, the parental strand as a template, and it builds a complementary or daughter strand. Now, remember how we said that the orientation of the strands is important here. Remember that the strands are arranged anti-parallel, and what this means then is that the new strand that is being built, the complementary daughter strand, has to be built anti-parallel to the parental strand. Well, in a replication fork, one of those parental strands is oriented in such a way that the DNA polymerase is able to just follow along behind the helicase. The helicase keeps moving along, separating the strands, and the DNA polymerase just keeps going, synthesizing a new strand just using the laws of complementary base pairing. The helicase moves, and the DNA polymerase moves with it. We call this continuous replication. And we call this strand that's being synthesized the leading strand. Remember, what you're having to build here are anti-parallel strands. So what about the other strand in the replication fork? Well, the problem here is that the new strand that has to be synthesized now has to be synthesized in the opposite direction from which the helicase is moving. And so what this means is that the synthesis machinery has to wait until the helicase has exposed enough of the DNA so that it can synthesize a complementary anti-parallel strand in the other direction. As such, we describe this as discontinuous replication. The reason being that one piece gets synthesized and then we have to wait for more of the DNA to be exposed before we can synthesize the next piece. And as such, this strand gets synthesized as a series of fragments. We actually call these Okazaki fragments after the pair of scientists that discovered them. So on one strand, we have continuous replication with the polymerase moving along in the same direction as the helicase. And that's our lead strand. But the other strand, it's happening in pieces. We call it discontinuous replication. We refer to this strand as the lagging strand because of the delay or the lag while we wait for the helicase to expose more DNA. Now, quick point, there's no actual real time lag here, but that's just the way that we refer to it. So this will continue on both strands, where the original parental strand is used as a template for, in one case, continuous production of a complementary daughter strand, and in the other, a discontinuous production of a complementary daughter strand. And this will continue until all that DNA has been copied. But let's step back a second here. Think about the primase. The primase is an RNA polymerase. It produces RNA primers that serve as the starting point for our DNA polymerase. In other words, our newly synthesized strands have RNA in them. Well, that can't work. We're synthesizing DNA here. We need to somehow get rid of this RNA. Well, here comes another all-important enzyme, a different type of DNA polymerase. This time, we call it DNA polymerase 1. And what it is going to do is scan along these newly synthesized strands looking for any RNA nucleotides. And when it finds them, it's going to cut them out and replace them with DNA nucleotides. Again, just using the laws of complementary base pairing. There's one more enzyme we need to think about, and it's called the ligase. When the DNA polymerase 1 has finished its work, a ligase enzyme comes in and ligates or ties together the final little gaps between where the DNA polymerase 1 has filled them in. And when that's done, so too is DNA replication. Now, this mode of replication, whereby we separate the two parental strands, use each one as a template for the production of a complementary daughter strand, we call semi-conservative replication. We call it this because each of the resulting what we call daughter DNA molecules has one original parental strand and one newly synthesized daughter strand. In other words, that original parental DNA is semi-conserved in each of the two new daughter DNAs. When all this is done, the cell will complete S phase, it will move into G2, and it can then prepare itself for mitosis, safe in the knowledge that when it finally splits, each of its two daughter cells are going to have all the information they need to function. Now, back to that ship. Is it the same ship? And if I'm replacing all my cells, am I the same person? I don't know.